All right, let's dive right into this explainer. We're going to map out the deep, underlying neurological mechanisms and the highly targeted pharmacotherapies of primary headache disorders, pulling straight from a masterclass level breakdown of Chapter 441. If you've ever heard someone dismiss a severe headache as just an everyday complaint, well, prepare to have your mind completely blown. We're talking exact genetic mutations, very specific neural pacemakers, and the literal revolutionary molecular treatments that are entirely rewriting how we handle these debilitating conditions.
Here's our roadmap for today. First, the anatomy of a migraine. Then, targeted pharmacotherapy. Third, the trigeminal autonomic cephalalgias. And finally, we'll wrap up with rare and situational headaches. Let's get right to it.
Section 1. Anatomy of a migraine.
So let's start by looking at the heavyweight champion of the headache world, the migraine. Look, this is not just a bad headache, right? It's a wildly complex, multi-phase neurological event that hits about 15% of women and 6% of men globally. The real trigger, it lies right here in the trigeminal-vascular system. You can see how sensory input from those pain-sensitive meningeal vessels passes straight through the trigeminal ganglion, synapsing on second-order neurons in the trigeminocervical complex. From there, the signal shoots right across the brainstem and projects up to the thalamus. Basically, this is the physical superhighway where those awful sensory processing dysfunctions, like that extreme, crippling sensitivity to light or sound, actually originate. Now, you probably know that about 20 to 25% of migraine patients experience this distinct phase called an aura. But what's actually happening in the brain? It's driven by a genuinely fascinating phenomenon called cortical spreading depression. Picture this. It is literally a slowly advancing wave of abnormal neural activity sweeping across the brain's cortex. This wave is exactly what causes those classic visual disturbances patients describe. You know, that slowly advancing visual scotoma with luminous edges that we call fortification spectra? It's a massive electrical shift, and it completely sets the stage for the pain that's about to follow.
Okay, so the absolutely crucial point here is the chemical cascade. Once those trigeminal nucleus cells fire up, they don't just sit there. They release these incredibly potent vasoactive neuropeptides directly at the vascular terminals. The big players? Calcitonin gene-related peptide, or CGRP, and pituitary adenylate cyclase activating polypeptide, or PACAP. The sudden release of these specific peptides is the primary trigger for that excruciating, throbbing pain and intense sensory sensitivity. Honestly, cracking the code on this exact chemical cascade has been the holy grail for developing modern treatments.
And what's just incredible to me is how perfectly the genetics map onto this mechanism. By studying families with familial hemiplegic migraine, researchers were actually able to pinpoint the exact ion channel mutations responsible. About half of these cases, which we call FHM type 1, stem from a CACNA1A gene mutation that completely alters a calcium channel. Then you have FHM types 2 and 3, and those trace back to very specific sodium, potassium, and neuronal sodium channel mutations, specifically in the ATP1A2 and SCN1A genes. These genetic discoveries are huge because they definitively prove that altered membrane excitability is what predisposes these patients to such severe auras and devastating attacks.
All right, moving on to Section 2, targeted pharmacotherapy.
So, now that we understand the problem all the way down to the receptor level, let's look at precision neuropharmacology. Because thankfully, the days of just throwing basic painkillers at a migraine are rapidly evolving. Look at the stark contrast between our primary acute treatments here. Triptans, which we've relied on for decades, stimulate the 5-HT1B and 1D receptors. And yeah, they're highly effective at arresting nerve signaling, but they also promote cranial vasoconstriction. Diptans, on the other hand, stimulate the 5-HT1F receptor. This means they act exclusively on neural targets in the central and peripheral nervous system, with absolutely zero vascular effects. This is a massive game changer for migraine patients who happen to also have cardiovascular or cerebrovascular disease.
But of course, that hyperspecific neural targeting comes with a pretty critical caveat. Because diptans, like the FDA-approved lasmiditan, act centrally, they can cause some heavy central nervous system side effects. In the clinical trials, roughly 15% of patients reported dizziness and another 6% reported somnolence. So patients taking lasmiditan absolutely must heed a strict, non-negotiable 8-hour driving restriction right after taking their dose. Essentially, it's a trade-off. You get unparalleled cardiovascular safety, but in exchange, you have to deal with a temporary central nervous system depression.
We're also squarely in the midst of the CGRP revolution. Remember that pain cascade we talked about a minute ago? Well, we now have drugs purpose-built to directly neutralize it.
Gepants, think rimogepant and ubrogepant, are oral small molecule CGRP receptor antagonists that we use for acute treatment. But even more remarkably, we have monoclonal antibodies like eptinizumab, eranizumab, fremonizumab, and galcanezumab. These target either the CGRP molecule itself or its receptor, and they're used for long-term prevention. I mean, they offer highly targeted relief with incredibly minimal side effects. It's completely changing the landscape of how we manage migraines.
Section 3. Trigeminal autonomic cephalalgias.
Okay, let's pivot for a second to trigeminal autonomic cephalalgias, or TACs. These are those excruciating, relatively short-lasting headaches that come with some really striking cranial autonomic symptoms like a watering eye or a stuffy nose on the exact same side as the pain. And the physical generators for these conditions are just deeply fascinating. If you look at a PET scan of a migraine, it shows activation in the dorsolateral pontine area of the brainstem. But check out the PET scan of a cluster headache. You see activation right in the posterior hypothalamic gray matter. Now, this is right near our human circadian pacemaker cells because cluster headaches are so remarkably periodic, often hitting at the exact same time every single day. This basically proves they're driven by a central premonitory pacemaker in the hypothalamus. It's a completely distinct mechanism from a standard migraine.
And differentiating these TACs is super crucial, mainly because their pharmacological responses are so incredibly specific. Take cluster headaches, which mostly affect men. They respond acutely to high-flow oxygen, and you can prevent them with verapamil. Though, a quick side note, that definitely requires careful ECG monitoring for heart block. Then you have paroxysmal hemicrania, which affects men and women equally and has its own incredibly specific drug response. And finally, SUNCT or SUNA, which present with these unique trigger profiles that require a totally different management strategy entirely.
Let's zoom in on paroxysmal hemicrania for a second just to see how wild and specific these treatments actually get. So these patients experience frequent, severe, short-lasting unilateral attacks. But here is the vital, non-negotiable diagnostic marker for this condition. It has an absolute, complete clinical response to a medication called indomethacin. Literally. If the headache doesn't completely resolve with indomethacin, then by definition, it is not paroxysmal hemicrania. It's a stunning example of how molecularly precise these headache categories have become.
And then there's SUNCT and SUNA, short-lasting unilateral neural G-form headache attacks. These are exceptionally rare, and they involve extreme stabbing pain right around the eye. But the defining feature here, the thing that really separates it from other neuralgias, is this bizarre, total lack of a refractory period between attacks. Imagine a patient gets a trigger touched and experiences agonizing pain. If that spot is touched again a split second later, the pain triggers immediately all over again. There's no cool down. Because of this non-stop excitability, patients can suffer up to 200 attacks a single day, which is exactly why we have to turn to nerve-stabilizing drugs like lamotrigine to finally calm that hyperactive neural pathway down.
Section 4. Rare and situational headaches.
Situational headaches are a great reminder of just how varied and intensely specific the nervous system's pain responses can be. Just imagine a patient coming into your clinic, deeply distressed, complaining of a moderate, generalized headache. But here's the catch. It jolts them out of sleep at the exact same hour every single night. It's literally acting like a biological alarm clock. What could possibly cause such a rigid, time-locked pain?
Well, this is the really curious case of the hypnic headache. It predominantly affects older adults, particularly women over the age of 60, and typically strikes a few hours after they fall asleep. Because of its unique pathology, it is highly responsive to a bedtime dose of lithium carbonate. Or, alternately, and this sounds totally paradoxical for a sleep disorder, taking a dose of caffeine right before bed can actually prevent the attack in about 1/3 of these patients. Crazy, right?
We also classify situational primary headaches based on highly precise physical triggers. There's primary cough headache, which is triggered suddenly by coughing or sneezing, essentially a Valsalva maneuver. Interestingly, this is wonderfully responsive to indomethacin. Then there's primary exercise headache, which has this throbbing, pulsatile quality, and it really requires patients to gradually build up their exercise intensity to avoid it. And there's also primary sexual headache, which can manifest as a dull ache that slowly builds, or as a sudden, severe, explosive pain occurring right at orgasm. But I need to pause for a highly critical warning here. We can only call these primary headaches once we are absolutely 100% certain they aren't secondary to something deadly. Anytime a headache is explosive, sudden, or tied to exertion, we must rule out life-threatening secondary causes first. For instance, did you know that 5 to 12% of cases of subacnoid hemorrhage are actually precipitated by sexual intercourse? Yeah. So whether it's a cough headache, an exercise headache, or a thunderclap headache, meticulous neuroimaging, like a CT or MRI, is an absolute, non-negotiable first step.
Which leaves us with this final thought for today. We've just mapped out how specific genes cause exact ion channel failures, how distinct hypothalamic pacemakers drive cluster headaches, and how custom-built antibodies can just neutralize CGRP pain cascades. So as we continue to decode these highly specific genetic and chemical pathways, you have to wonder, will the mystery of the headache soon be entirely solved? Are we actually looking at a future where nobody ever just has a headache, but rather receives a precisely diagnosed, perfectly treatable molecular event? It's an absolutely fascinating frontier, and it completely reframes how we view human pain. Thanks for joining me on this explainer. Keep digging into the research, stay curious, and I'll catch you next time.